Hello guys, welcome to Bioguru. Today we will discuss about the prokaryotic cells. The cells having primitive types of nucleus are called prokaryotic cells, just like bacteria, blue-green algae, mycoplasmas and PPLO. PPLO is pneumonia-like organisms. Now the prokaryotic cells are generally smaller and multiply more rapidly than the eukaryotic cells. तो prokaryotic cells है bacteria इनकी लाइफ बहुत छोटी होती है, इसलिए रैपिडली डिवीजन करेंगे, डिवाइड करेंगे, नंबर ऑफ कॉपीज बनाएंगे. दे मे वेरी ग्रेटली इन शेप एंड साइज. जो बैक्टीरियाज हैं उनके भी अलग अलग शेप हैं, अलग अलग साइज हैं, बट देयर आर 4 बेसिक शेप्स. सो 4 बेसिक शेप्स आर, फर्स्ट इज बैसिलस, दैट इज रॉड लाइक. सेकंड इज कोकस, दैट इज स्फेरिकल. देन विब्रियो, दैट इज कॉमा शेप, एंड स्पाइरलियम, दैट इज स्पाइरल शेप. Then organism and their range. Typical eukaryotic cell size क्या होगा? 10 micrometer to 20 micrometer. Typical bacterial cell 1 to 2 micrometer. PPLO 0.1 micrometer. और viruses बहुत छोटे होते हैं, 0.02 to 0.2 micrometer. Generally prokaryotes have a cell wall surrounding the cell membrane. And the fluid matrix filling the cell is cytoplasm. जो plasma membrane के अंदर का जो fluid है, जो matrix, that is called cytoplasm. And there is no well-defined nucleus because they are prokaryotic cells. They are prokaryotic organisms, और prokaryotic cells हैं, prokaryotic organism हैं, so they have primitive nucleus. And the genetic material is basically naked. Naked means they are not surrounded by any membrane or any boundary. In prokaryotes, the genomic DNA stands for single chromosome, circular DNA. देखिए वैसे तो प्रोकैरियोट्स में क्रोमोसोम्स नहीं होते, यूकैरियोट्स में क्रोमोसोम्स होते हैं. एक्चुअल में यूकैरियोट में जो क्रोमोसोम होंगे तो डीएनए के साथ बहुत सारे हिस्टोन एंड नॉन हिस्टोन प्रोटीन्स भी होते हैं, बट हिस्टोन एंड नॉन हिस्टोन प्रोटीन्स आर एब्सेंट इन द प्रोकैरियोट्स, इसलिए प्रोकैरियोट्स में क्रोमोसोम्स नहीं होते. बट यहाँ पे एक सिंगल जो क्रोमोसोम है, जो डीएनए है, वो कंडेंस फॉर्म में है, इसलिए उसे हम सिंगल क्रोमोसोम भी कंसिडर कर लेते हैं. ओके, so in addition to genomic DNA, many bacteria have circular DNAs outside the genomic DNA called as plasmid. जो मेन डीएनए है, जो जीनोमिक डीएनए है, उसके अलावा एक्स्ट्रा एडिशनल क्रोमोसोम या एडिशनल डीएनए है, इट इज कॉल्ड प्लाज्मिड. सो प्लाज्मिड्स आर एक्स्ट्रा क्रोमोसोमल सेल्फ रेप्लिकेटिंग क्लोज सर्कुलर डीएनए, एंड ये बैक्टीरियाज को कुछ एक्स्ट्रा कैरेक्टर्स प्रोवाइड करते हैं. देन देखिए प्लाज्मिड्स, एडिशनल स्मॉल सर्कुलर डीएनएस. ओके, द प्लाज्मिड डीएनए confers certain unique phenotypic characters to such bacteria, just like antibiotic resistance. And nuclear membrane is found in eukaryotes but not in the prokaryotes. And but ribosomes are present in both prokaryotes as well as eukaryotes. Mesosomes are the characteristic of the prokaryotes because mesosomes are found only in the prokaryotes. These are not found in the eukaryotes. So ribosomes आप जानते हो prokaryotes and eukaryotes दोनों में ही मिलते हैं. तो mesosomes क्या थे कि extensions into cytoplasm are called mesosomes. So membrane extensions. Now cell envelope and its modification. जो prokaryotes हैं उसमें कुछ modifications हैं, cell envelope उनकी बात करेंगे. So bacterial cell have chemically complex cell envelope. The cell envelope consists of a tightly bound 3-layered structure. Outermost glycocalyx followed by the cell wall and then plasma membrane. The cell envelope consists of glycocalyx, cell wall and plasma membrane. Although each layer of the envelope perform distinct function, they act together as a single protective unit. तो combined form में together they form a protective unit. And bacteria can be classified into 2 groups based on their chemical composition of envelope and response to staining. The staining procedure developed by the gram. तो gram positive and gram negative आपने 2 bacteria के बारे में सुना हो. So bacteria based on response to staining, gram positive and gram negative. So bacteria that take up the gram stain is called gram positive bacteria, and bacteria that do not take up the gram stain is called gram negative bacteria. So glycocalyx differs in composition and thickness among different bacteria. जो ग्लाइकोकैलिक्स है उसकी थिकनेस, उसका कंपोजिशन, ये ग्राम पॉजिटिव एंड ग्राम नेगेटिव बैक्टीरियाज में अलग अलग होता है. सो इट इज द envelope of the gram positive bacteria, then it is the envelope of gram negative bacteria. Now glycocalyx, it could be a loose shape called the slime layer, and it may be a thick and tough layer called the capsule. The cell wall determines the shape of the cell because जो cell wall है वो outermost covering है, it is very stiff, hard. So cell wall provides a strong structural support to prevent the bacterium from bursting or collapsing. And then what determines the shape? That is, it is cell wall. So the plasma membrane is semi-permeable because जो plasma membrane है वो semi-permeable है, वो सारी चीजों को allow भी नहीं करेगा और सारी चीजों को restrict भी नहीं करेगा. This membrane is structurally similar to that of the eukaryotes. A special membranous extensions of plasma membrane into the cell cytoplasm is mesosome. क्योंकि जो plasma membrane का अंदर की तरफ जो extension था उसको हमने क्या कहा था? Mesosomes. So extensions are in the form of vesicles, tubules and lamellae. So functions of mesosomes: cell wall formation, DNA replication and its distribution to daughter cells, helps in respiration, secretion processes to increase the surface area of the plasma membrane, that is absorption of nutrients, and enzymatic content. In some prokaryotes like cyanobacteria, there are other membranous extensions into the cytoplasm called chromatophores because chroma term is used for the color or pigment. That's why they contain pigments. Then bacterial cell may be motile or non-motile. Motile का मतलब है जो move कर सकते हैं, non-motile जो move नहीं कर पाएंगे. If motile, they have thin filamentous extensions from their cell wall called the flagella. And bacteria show a range in the number and arrangement of flagella. देखिए अगर मान लीजिए सिंगल है तो मोनोट्राइकस हुआ, चारों तरफ हैं पेरीट्राइकस है, लोफोट्राइकस है, एम्फीट्राइकस, तो उनके अरेंजमेंट के बेसिस पर, उनके नंबर के बेसिस पर भी उनको क्लासिफाई किया जाता है. सो इट इज द स्ट्रक्चर ऑफ फ्लैजिलम. सो बैक्टीरियल फ्लैजिलम इज कंपोज्ड ऑफ फिलामेंट, हुक एंड बेसल बॉडी. बेसल बॉडी जनरली वो मेन पार्ट है जो प्लाज्मा मेम्ब्रेन में एम्बेडेड होगा. Then hook basal body and filament को connect करेगा, and filament is the long extension. So the filament is the longest portion and extends from the cell surface to the outside. Besides flagella, villi and fimbriae are also surface structures of the bacteria but do not play a role in the motility. मतलब वो movement में help नहीं करते. The villi are elongated tubular structures made up of special proteins. So longest extension is filament. The fimbriae are small bristle-like fibers sprouting out of the cell, and function क्या है इसका? Attachment. So in some bacteria, they are known to help attach the bacteria to rocks in streams and also to the host tissues. तो ये attachment के लिए, anchoring के लिए use होते हैं. Ribosomes. Because prokaryotes में ribosomes होते हैं, and prokaryotes have 70s ribosomes. So in prokaryotes, ribosomes are associated with the plasma membrane of the cell. They are about 15 nanometer to 20 nanometer in size, and ribosomes. अब देखिए जो ribosomes है made up of two subunits, 50s and 30s. 70s को break किया गया 50 plus 30 में, which when present together form 70s prokaryotic ribosome. Ribosomes are the sites of protein synthesis. That's why they are called protein factories. तो राइबोसोम का साइज क्या था? 15 nanometer 20 nanometer. Several ribosomes may attach a single mRNA and form a chain called polyribosome or polysome. अगर एक RNA strand पर बहुत सारे राइबोसोम आके अरेंज हो जाते हैं, that structure is called polysome or polyribosome. The ribosome of polysome translate the mRNA into protein. What is the meaning of translation? Formation of proteins from RNA is called translation. Inclusion bodies, reserve materials in the prokaryotic cells. कुछ मटेरियल है, फूड मटेरियल या अदर मटेरियल, उनको रिजर्व किया जाएगा स्टोर करने के लिए, are stored in the cytoplasm in the form of inclusion bodies. The inclusion bodies are not bounded by any membrane system and live free in the cytoplasm. तो freely float करेंगे. They are not bound with the any membrane. Phosphate granules, then cyanophycin granules and glycogen granules are the examples of inclusion bodies. Gas vacuoles are found in blue-green, purple and green photosynthetic bacteria. So now some MCQs. The structure of the prokaryotic cell that separates the interior of the cell to the exterior of the cell. Cell wall, nucleoid, cell membrane or slime layer. It is cell membrane. That's good. The ribosome of polysome translate the mRNA into carbohydrate, lipids, proteins or sugars. Yeah, proteins. Genomic DNA of bacteria is naked, linear and double-stranded, membrane-bound, circular and double-stranded, naked, circular and double-stranded, or naked, linear and single-stranded. It is naked, circular and double-stranded. Circular DNA. Outside the genomic DNA in bacteria is called plasmid, viroid, virion or prion. Yeah, it is plasmid. Reserve materials in prokaryotic cells are stored in chromatophores, inclusion bodies, mesosomes or plasmids. Inclusion bodies, that's good. Thick and tough glycocalyx present outside the bacterial cell wall is called slime layer, capsid, capsule or papilos. Yeah, capsule.